Hey you guys, we are looking at the FRQ on bed bugs and experimental design. And so I believe this FRQ was pretty straightforward, and it was more about kind of keeping your game together, reading the information and processing it and coming to a conclusion. And so they just gave you data to break down, and none of the questions were really too out there. So let's look at the data they gave us. On the y-axis, it is the percent survival. And so as you increase on the y-axis, you have a greater survival rate. And on the x-axis, you have 5 different strains of bed bugs. Each bed bug strain had different genes deleted. And so if we look at strain 2, this community or this population of bed bugs has the P450 gene deleted. And so we're looking at the effects of deleting genes on bed bugs. And so here we know the first question here says identify the strain in the experiment, the control strain in the experiment. And we know that that's the first one because here, 1, 2, 3, all of these genes were left intact. Those are the wild type genes. So this is the bug that you would find in nature. Everything else has been changed. There's been an independent variable applied. Some gene has been deleted. And so it only asks for the identification. You don't need to justify your answer there. What it does want you to justify is the claim that ABC8 is effective at providing resistance to this insecticide. And so the justification, if you look, this is where we're going to be using our error bars or our confidence intervals. And so here we have within 95% confidence, we can say that the mean survival of the second strain of bugs is within 40%. And so here we're saying that the ABC8 strain is the most effective. And why is that? Well, if we look at where it was applied in both strain 3 and strain 5, both of these scenarios, the mean number, including the error bars, are less than the mean of the control, including the error bars. So, since the error bars do not overlap at all, that means that there is statistical significance to this data, that there is an effect, something is happening, something has been applied that's affecting the way that this strain is interacting with the beta cyflurin. So that is the first part of this. And so the second part, P450 encodes an enzyme that detoxifies insecticides. ABC8 encodes a transporter protein that pumps insecticides out. CPS encodes external structural protein located in the exoskeleton, basically reducing the absorption. So explain how a deletion of both 450 and ABC8 results in lower survival. Basically, this is, they gave you the answer in the question. Bed bugs, you're kind of double whammying the bed bugs with the P450 and ABC8. You are detoxifying the pesticide, and you are transporting it out. So the combination of these two is far more effective than just reducing absorption. When you reduce absorption, you are still letting some of the insecticide in. So here you are detoxifying it and pumping it out, which is definitely more effective than just allowing less in. So here's a sample example, and I hope that helps.